Hello everyone, I'm Manaswi. I will be presenting work that I did during my PhD. I basically studied the impacts of woody plant invasion in the Shola-Grassland ecosystem of the Nilgiris Biosphere Reserve. I will first introduce this ecosystem since I understand many of you might be unfamiliar with this. The Indian subcontinent has several types of ecosystems, from deserts to savannas to rainforests. Along the west coast of India lies the Western Ghats biodiversity hotspot, a step-like mountain chain with different ecosystems at different elevations. If we zoom in further, we can see one such system in the Nilgiri Biosphere Reserve called the Shola-Grassland ecosystem. This ecosystem is a biphasic mosaic of forests, locally called sholas in the sheltered slopes and valleys, and grasslands in the exposed slopes and hilltops. These mosaics are extremely biodiverse ecosystems, providing habitats to many species of conservation interest. Some of these species are endemic to the ecosystem. There are also many water reservoirs in the Nilgiris. Many of them are sources for rivers, important rivers in the Indian subcontinent. So it's a very important place for us humans as well. However, the Nilgiri grasslands are being invaded by three nitrogen-fixing woody species. In the photo on top here, you can see that one of them, the 𝐒𝐴𝐺𝐚-𝐆𝐫𝐚 from Australia, has invaded this area surrounding the Shola forest patch. The other two species are 𝐒𝐡𝐨𝐥𝐚 copareus or Scotch broom and 𝐒𝐢𝐥𝐚 europaeus or gorse from Europe. Since 1973, the Nilgiri grasslands have decreased in area by 66%, and most of this loss is due to invasion. The forest department is trying to manage this by manually removing the invasives. However, this has been mostly unsuccessful. The area highlighted here was cleared of acacia invasion in 2012, but as you can see, the acacia is back. The invasives may be transforming the system and creating conditions which prevent successful restoration. Since the woody invasives are invading only the grasslands and not the Shola forests, I would like to mention a couple of points about the grasslands in our study area. The grasslands are mostly dominated by grasses which follow the C4 photosynthetic pathway. These grasslands also have very high diversity of forbs, which are basically herbs that are not grasses. Forbs also form a part of the diet of herbivores and provide food and habitat to many invertebrate species as well. Many forbs have underground storage organs from which the plant community can recover after a disturbance, such as a fire. Finally, these grasslands also have extremely low abundance of native nitrogen-fixing plants. This brings me to my research questions. We first asked, how do the invasives impact soil inorganic nitrogen cycling? We next asked whether invasives are impacting nitrate outflow from the ecosystem. And finally, we asked whether the invasives change the native grassland community and functional composition. In each sampling location, this is what I did. I collected a fresh soil sample, which is till 10 centimeter of soil depth. The nitrate and ammonium concentrations in this sample tell us how much inorganic nitrogen was present at the time of collection. I took another soil core, put it in a PVC pipe, which was covered from the bottom so that plant roots cannot enter. So changes in the soil within this pipe is because of microbial activity alone. The pipe was installed back in the field for about 2 months, which is the T in the equation here. At the end of the 2-month period, again the inorganic nitrogen concentration was measured. The difference in the concentration would be due to microbial activity alone, which tells us what is the rate of the nitrogen mineralization in this soil. To measure nitrate outflows, we chose 9 different catchments with different intensities of invasion by 𝐒𝐴𝐺𝐚-𝐆𝐫𝐚. Approximately twice a month, we collected water samples flowing out of each of these catchments. So what do the results look like? Here on the y-axis, we have the inorganic nitrogen concentration. The purple line is for the grasslands, and the other three colors are for the areas invaded by the invasive species. We see that total inorganic nitrogen is highest in the starting of the dry season and remains high during the dry season. This is probably because plant growth is not happening during this season, and nitrogen mineralization rates are positive in the dry season. Further, most of this increase in the inorganic nitrogen is driven by increases in soil nitrate concentrations, with invaded areas having much higher nitrification rates. Nitrate is an extremely mobile form of inorganic nitrogen, which basically means that it moves easily in ecosystems. So the question arises as to what happens to all this extra nitrate that is being introduced by the invasors. In this graph, I have plotted the nitrate concentration in stream outflow as a time series, with gray boxes denoting the period of the main rainy season. As you can see, the nitrate outflow is higher in the rainy season, and it seems to be higher from the more invaded catchments, the darker lines here. So we decided to focus next on the rainy season alone, the results of which you will be seeing in the next slide. The x-axis here is the rate of water outflow from each catchment. On the y-axis is the nitrate concentration in the stream. The 9 panels are basically the 9 different catchments ordered by increasing invasion by 𝐒𝐴𝐵𝐚. We see here that the outflow of nitrate is higher with increasing water outflow. And this outflow of nitrates is even higher in catchments with higher 𝐒𝐴𝐵𝐚 invasion than in catchments with lower 𝐒𝐴𝐵𝐚 invasion. Next, we looked at how the vegetation in the woody understory differs from the vegetation in the grasslands. I included 2 exotic tree species which have been planted in the Nilgiris but are not invasive yet. These are Eucalyptus globulus and Pinus petula. Just as an example, this is what I mean by the understory. This is the understory of a pine plantation from our study area. In this graph and the upcoming graphs, the green color is for the grassy patch and the black color is for the woody understory. We see that the total species richness is lower in the woody understory in all the cases in our study area. This is true for both the grass richness and the forb richness. In particular, C3 grasses were dominant in the woody understory, while in the grassy patches, C4 grasses were dominant. Many of the C3 grasses in the woody understory are invasive grasses themselves, suggesting that woody invasion is facilitating secondary invasion by exotic grasses. So, summarizing, we see that in the Nilgiri grasslands, the invasive and exotic species are affecting grassland composition by decreasing species richness and by shifting the dominance from C4 grasses to C3 grasses, many of which are invasive grasses. Further, the invasives increase soil nitrate concentrations and soil nitrification rates and cause greater outflow of nitrates from the ecosystem. What are the implications of these results for restoration efforts? Our results point towards the need to reintroduce native forbs and C4 grasses in restoration activities. Further, the elevated soil nitrogen levels can facilitate further invasion, suggesting that restoration efforts may need to decrease soil nitrogen levels. Finally, continued invasion can cause eutrophication in the downstream water reservoirs of the Nilgiris, pointing to the need to manage for nitrate export from invaded areas. With this, I would like to stop my presentation and thank you for your attention.